Protein synthesis time! The central dogma is basically the flow of how the blueprint in your DNA becomes you. DNA contains instructions for making the protein, which are copied to make RNA in transcription. RNA then undergoes translation. Its information is used to build a polypeptide. Now let's go into the details. Transcription. In the nucleus, an enzyme called RNA polymerase attaches to the DNA and finds a specific genes it needs to copy. It unwinds that small section of the double helix and separates the two strands. RNA polymerase reads the template strand of the DNA. It grabs free-floating nucleotides and pairs them up with the DNA template. Following base pairing rules, thymine pairs with adenine. Remember though, RNA does not contain thymine, so it's actually uracil that pairs with adenine. Guanine pairs with cytosine, cytosine pairs with guanine, and so on. This new single strand of RNA is called pre-mRNA, a draft copy of the gene. And by the way, M in mRNA stands for messenger, which you will see why. mRNA processing. Before mRNA leaves the nucleus, it has to be edited and protected. There are two types of sections in the new mRNA strand. Introns, which are the junk sections that don't code for the protein. These introns are discarded. Exons, in which X stands for expressed, do code for the protein. They are stitched together. Now, the mRNA strand gets some protection. A methyl cap is added to the 5' end, allowing the mRNA to leave the nucleus and helping the ribosome with finding it later. A poly A tail or a long chain of adenine bases is added to the 3' end, acting as a shield to protect the mRNA from being broken down by enzymes called exonucleases. With full protection, the mature RNA strand is ready to leave the nucleus and go to the cytoplasm. Translation. Now that the mRNA is in the cytoplasm, a ribosome, which is the factory of protein synthesis, clamps onto the mRNA strand and starts creating codons. A codon is basically a group of 3 nucleotides. Each codon specifies 1 amino acid. Now we have something called transfer RNA or tRNA. At the bottom, there's an anticodon that pairs with a specific mRNA codon. At the top, there's an amino acid that matches the codon. Lots of tRNAs like this float around in the cell, but only the one that matches the codon is used. The process starts when the ribosome reads the start codon AUG. Then the tRNA with the anticodon UAC floats in, carrying the amino acid methionine and locks into place. Then the ribosome moves to the next codon. The matching tRNA floats in, and the ribosome joins the first amino acid to the second one. Notice, the first tRNA is now in the P site. The second tRNA is in the A site. As the ribosome moves forward, the empty tRNA moves to the E site to exit. The other tRNA moves to the P site, and the process repeats, building a long chain of amino acids. Eventually, the ribosome reaches the stop codon like UAG, UAA, or UGA. The release factor will enter the A site and translation is terminated. The new polypeptide is complete. It folds into a 3D shape, becoming a functioning protein. And that is protein synthesis. Thanks for watching.